So in essence, what I'm doing is trying to cut down on the inefficiency in the first stage of drug discovery by miniaturizing the process and making it much cheaper and possibly available to academic labs and even individual patients. So this is a standard tissue culture plate that's used for growing cells. Pharmaceutical companies use these where you get one test per well. And what we're working on is to get thousands of tests per well by printing the different drugs inside of the wells.
I culture them for 24 hours,
Ah.
so I'm going to look at them underneath the microscope.
So there's a whole field of science called lab on a chip, which is trying to miniaturize laboratory activities. And there's sort of two approaches to that. One is called microfluidics, and another approach is the microarray approach, which is what I'm taking, where you put all the different materials on a surface in a microarray, and then you can put one solution on top of all of that. But the general idea is to try to do what a laboratory does using a chip. This is a picture of a liposome microarray. And so each of these red dots in principle would have a different drug in them. And what we do is then we grow cells on them.
Because they're so small, we can fit many different types of drugs on the same surface that are encapsulated in the liposomes.
And so my idea is kind of twofold. First, it's to lower that cost, the initial cost of screening all those drugs. And at each stage, there's approaches being taken to lower that cost. And the second approach is by lowering that cost, you can make those technologies available for personalized medicine. So there's a big movement also in the FDA. They know this, that you can't just make a rule for everybody and say this drug will be given to every cancer patient. You have to figure out which one's going to work on an individual. So there's genetic approaches. The approach I'm taking is to take the cells, the cancer cells from the patient, and miniaturize an assay so that you can test as many different drugs as possible on that patient's tumor. I'm sure in 50 years, you'll have something like the tricorder in Star Trek, which is their whole lab, and they go inside, they detect life signs and everything. So that's kind of the hope that we'll be able to do what you normally need a laboratory to do, but on a computer chip.